Good evening, students. Today we will revise blood. Okay, 5 points I'll try to cover. So, introduction is blood is a tissue fluid. It contains cells and plasma. The cells are red blood cell, white blood cell, platelets. Plasma is a fluid part and it contains plasma proteins, namely albumin, globulin, and fibrinogen and clotting factors. So, what is serum? Serum is the plasma devoid of the clotting factors. This is the first point. Second point is red blood cell. Red blood cell, the development and maturation is called erythropoiesis, and it occurs in stages starting from hemocytoblast, pro-normoblast, early normoblast, intermediate, late normoblast, reticulocyte, and RBC. So hemoglobin appears at early normoblast, nucleus disappears at late normoblast. And the factors stimulating are erythropoietin is an important hormone. It is secreted by the peritubular capillaries of kidney in response to hypoxia. And thyroid, testosterone, these will stimulate the erythropoiesis. Estrogen inhibit erythropoiesis. Vitamin B12 and folic acid, they helps in the maturation of the DNA, and the lack of this leads to megaloblastic anemia. Third point is WBC. So WBC, we have granulocytes, A granulocytes. Granulocytes are neutrophil, eosinophil, and basophil. A granulocytes are lymphocytes and monocytes. So here the role of WBC in immunity to be discussed. So what is immunity? Ability of the body to defend against microorganism. There are two immunity: humoral immunity, cell-mediated immunity. Humoral immunity is mediated by B lymphocytes. Cell-mediated immunity is mediated by T lymphocytes. So in humoral immunity, after the antigen presentation, the helper T cells secretes interleukin that activate the B cell to plasma cell, and they secrete immunoglobulins, and these immunoglobulins attacks the antigen by agglutination, precipitation, neutralization, and complement activation. And in cell mediated immunity, the specialized antigen presenting cell, macrophage, present the antigen to the T cells, and there are two T cells, CD4 and CD8 T cells. CD4 T cell recognize antigen with MHC2 complex, and CD8 T cell, that is cytotoxic and suppressor T cell, they recognize the antigen with MHC1 complex. Now here the cytotoxic T cell is direct cytolysis. It kills the antigen by forming perforin proteins. And the role of helper T cell, as already discussed, it helps the B cells to differentiate into plasma cells. And regarding the platelets, so the fourth point is platelets. So platelets forms platelet plaque, which is an essential step in hemostasis. First it gets attached to the damaged vessel wall, then it gets activated and it secretes chemical substances that makes the aggregation of the platelet. 3A: addition, activation, and aggregation. This forms the platelet plaque. And a platelet plaque is the step in hemostasis. So what is hemostasis? Arrest of bleeding. It occurs in 3 steps. First one is vasoconstriction, second one is platelet plaque, third one is clot formation. Now we'll see what is clot formation. Clot formation is formation of the fibrin is called clot formation. It makes the temporary plaque into a permanent plaque, and it occurs by the activation of the clotting factors. There are 13 clotting factors. So there are two pathways, intrinsic pathway and extrinsic pathway. Intrinsic pathway gets activated when the blood is exposed endogenously to the damaged collagen. Activation of factor 12, then 11, then 9, then 10, then factor prothrombin converted to thrombin. Thrombin converts fibrinogen to fibrin. So fibrin is formed. In extrinsic pathway, the activation occurs outside the blood vessel, that is release of tissue thromboplastin that activate factor 7 to 7A, and this 7 factor will activate factor 10, and this activate prothrombin to thrombin, and thrombin will convert fibrinogen to fibrin. So the ultimate result is formation of the fibrin, and it is stabilized by the fibrin stabilizing factor. And the final fifth point is the applied aspects. What is anemia? Decreased RBC or hemoglobin content. Most common anemia, iron deficiency anemia. It is a microcytic hypochromic anemia. In immunity, you have to remember autoimmunity, that is antibody produced against the body's own tissue. Example, myasthenia gravis. And third point is the clotting factors in applied aspects. So what is clotting disease? A coagulation disorder is deficiency of clotting factor 8 leads to hemophilia. and low platelet count leads to thrombocytopenic purpura. So clotting factor defect leads to clotting diseases or coagulation disorders, and platelet defect leads to bleeding disorders. So these are the points to be remembered. Thank you.